All right, here we are. We're going to start our class right now, and we have our test on Thursday. So one thing you should do is go over the old exam and all kinds of stuff. It's going to cover everything we dealt with so far in our course. Uh-oh, that's not good. And then let's make sure that the recording is still going, because sometimes that interrupts the recording. And then, let's make it a big image and say that we also have that make drawing assignment. So that's due, and that'll be part of our exam, of course. So that's where we're at. So what we're going to do is, and then I've talked about everything I really want to talk about that will be on our first exam. If we have time today, I will just add new material that will not be on our exam. And I'm excited to do that, right? And so, but we can take as much time as people want at the start of class or any time within the class meeting to ask questions that are, what about the exam? What about this? What about that? And so let's do that. And then the other thing I want to say is that, people are also starting to work on their term paper. And, and understand that this week, though, you're really focused on the exam. So we got to get that going. But as soon as we meet again next week, we need to deal with that. So with regard to the exam, remember that it's like the previous exam. So that was 50 two-point questions. Sometimes the 2 points get glued together because I'll say, explain something like how to make a transgene and make a mouse with muscle that glow. And so I'll say that's 4 points right there, So sometimes I chain them together a little bit, but they're always isolated points, and you're answering questions trying to hit the points like that. The questions will be very clear, and you want to read the words carefully, because if it says, give 3 examples, you have to do that or you'll start losing points. I also really am reluctant to say, oh, this is worth half a point. I try really hard to build questions that you either get right or wrong, and I don't have to, there's another half point there, right? So think of it like that. It'll be a boom, boom thing, and you don't want to waste your time over explaining things. You can, that's fine, but I've talked about that several times. The only issue with that is you start running out of time, and you're like, I'm only on question 3 because I wanted to share everything I do about the estrogen receptor. And I say, the first question on the exam is just, what are the 3 domains of the estrogen receptor, and which one has zinc? Like that, right? So there's 2 points there. 3 domains, and one of them is got a zinc atom in it, like that. Okay, so that's that, and indeed, this is a hard course, right? So we're learning, but there aren't any tricks on the exam. So if you start looking at the exam going, oh, is this a trick question? No. And then remember that there will be a few points on the exam that were in the book, but were never covered in the class. And those are more obvious points, like a bold term that's clearly a big term that I never used, or a box, a whole page talking about a method. What are the 2 points on that page? I'm talking about that method. Yep. So let's start with that and see if people have any questions they want to. And I should mention that in answering questions, it's possible that new material will come up today, right? So I don't plan to, give you any new material, but there's, when I start answering questions, I might describe another example, and I'm like, now that's part of our course. So we likely will have to learn some, make sense? Okay, so let's see if people have questions. Yes?
I do in chapter 7, there was a bold letter under histones and epigenetic inheritance, and it was epigenetic inheritance, and it describes the transmission of information from parent to progeny that is contained within the sequence of DNA. How does that happen?
Yep. So there's a word, epigenetic, and it actually has 2 definitions. The first definition came out from a geneticist in the 1940s, and it really was the first time. At that time, it was theoretical. Scientists did not know that DNA was the primary heritable molecule, and that DNA held physical genes, like that, right? So this old-time geneticist used this word epigenetic, and his idea was essentially that a cell would have more genes than it was using at any one time. And we know that's super true, right? They just didn't know anything about DNA or genes. They didn't know about that there was genomic equivalence. We know now that every cell in our body has all the genes, it's just not using them. So that's the original definition of epigenetics. Almost no one talks about that today because it's just so obvious that that's true. There are a variety of ways that epigenetics occurs. The most common one is regulation of transcription. So that's why our course focuses so much on that. There's another one, there's several, and the book talks about that in chapter 7. It's talking about gene regulation. So it's talking about the concept of epigenetics. There's another specific example of epigenetics that scientists didn't know how to study very well until about 15 years ago. And then they developed methods. And so science is like that. There's currently topics that are most exciting because scientists came up with methods to tackle those, and so there's a lot of new data. And that is DNA methylation. So DNA methylation is a type of epigenetics. DNA methylation affects gene expression. And so far, I think it only affects gene expression by repressing it. And if there's certain DNA methylation occurs, the gene cannot be expressed. So that's what people usually mean when they say epigenetics today, right? So then it's DNA methylation. And so that gets to your question, Cam, and that is that in some organisms, those patterns of DNA methylation change depending on the environment. And I mentioned one example of that is a queen ant and worker ant. They're identical twins. The difference is their environment. The queen ant, when it was a larva, was fed a little bit different food that caused differences in DNA methylation. So that's the big difference between a queen ant and a worker ant. And so that's an example of how the environment can affect DNA methylation. And students are excited to learn about that. The internet is full of people writing about that because it's part of the DNA, but it's not a change in the letters A, G, C, T. It's just whether the C gets methylated or not. And C is the only one that gets methylated. And if it gets methylated, it could be involved in gene repression. So next thing is that in some examples, that pattern of DNA methylation can get inherited from one generation to the next. And people are super excited about that. And there are very specific examples in certain animal species where that occurs. So that's what the author is talking about, is they're talking about especially DNA methylation as a potential example of where something can get inherited from one generation to the next. Now, it's not permanent inheritance because the DNA methylation could get reversed. It's not permanent inheritance like the A, G, C, T letters. Those can only change with a mutation, and then that's permanent, right? Like that. So that's what they're writing about there, is it's really zoomed in on that. And that's one of our examples. We want to know that about remodeling chromatin, because people usually stick that DNA methylation in as an example of remodeling chromatin. So it's one of our 3 examples that we want to know about, but I've really just said that. We don't need to know the structure of the nucleotides and where the methyl group gets attached, but just that it's DNA methylation and it can inhibit gene expression. And our favorite example in our course is differentiation. Differentiation involves DNA methylation. And then our second favorite example is how the scientists could turn any cell, even a differentiated cell, into a stem cell. So that's an experiment they can do in lab. And of course, what they have to do in lab is they have to demethylate those Cs to allow every gene to be expressed again. So we learned in the first videos, the chondrocyte, a differentiated cartilage cell, it's got all the muscle genes, but it's methylated up. And so it cannot make muscles. But scientists can reverse that in a lab, and they can take a differentiated stem cell, a differentiated chondrocyte, cartilage cell, and turn it back into a stem cell, and eventually make muscles. That's really what that MyoD is doing in the precursor, is it's changing the fate of the cell by changing the epigenetics, right? Changing the gene expression pattern. Yep. Great. And yeah, I always bring my textbook, you know, so people can just do like that. That's fantastic. Yep. Other questions people have? Yep.
I have a question about the G-coupled protein receptor. Is the G protein bound before the ligand, or does it bind to it after the ligand binds to the G protein?
It varies.
Okay.
So, and that's a detail. So in the G protein coupled receptor pathway, we have the G protein coupled receptor, and when it binds to the ligand, it's going to get activated, and it's going to directly cause the G protein to exchange GDP for GTP. And in some cases, the G protein is already bound. I think in the majority of cases, it's not already bound.
Because I don't remember because what I read what we said, but I remember at one point something was said that like the G protein isn't activated until it converts its GDP for GTP. So I didn't know if it like was on its own timeline or if it was the binding of the ligand that immediately caused it to have that change, and then the alpha gets.
So people would say that when the G protein gets activated, they would say it has exchanged its GDP for GTP.
But it's a binding of the ligand to the receptor that causes it.
Yeah, that
Okay.
leads to that. Yeah, and because ultimately, right, it's the G protein that's holding on to GDP, and to activate, it has to exchange that for GTP, and it does that by binding to the receptor. Now, I don't really try to emphasize that because it's just an automatic thing. When it binds the receptor, it just automatically exchanges GDP to GTP. The two things we want to emphasize about that are that it's always bound to one or the other, and when it is activated, it's not an enzymatic activity. It's just a binding activity. I'm going to let go of this GDP, and here's some GTP in the cytoplasm that I will bind immediately. I'm going to exchange those, not enzymatically, because that's the activation, and in contrast, it's the inactivation that requires the enzymatic activity. That's not an exchange, that's where that GTP gets hydrolyzed to GDP in a chemical reaction that is enzymatically catalyzed by the G protein. Yep.
Is that true for heterotrimeric G proteins as well as monomeric G proteins?
Yes.
What is the name? So it activates. So like when the CAMP activates a protein kinase A, the picture then shows just one part of it. That is still like that's just a piece of protein kinase A, or is that something? It just labels it as a C. What does that mean?
Catalytic subunit, yeah.
Catalytic.
So yeah, so that protein kinase A that gets activated by cyclic AMP, that book shows it pretty accurately. It's a dimer, and it's when it's activated, it's a tetramer when it's activated, because it has some subunits that are inhibitory subunits. But that's diving into the details, and I mentioned that in class. For us, it'd be great to say that when cyclic AMP gets made, one of its jobs, and an important job, is to activate protein kinase A. And then protein kinase A is activated and will phosphorylate targets like the enzyme that breaks down glycogen. And again, we don't need to know the name of that enzyme, just the idea that we've got lots of polymers of glucose in each of our cells. It's called glycogen, and there's an enzyme that just cut it, break down to individual glucose. Little side note on that, but before I go to that side note, then the other target that we talked about in a different story is Krebs. Yeah. So a little side note on glycogen that's just a sweet story to know about, and that is that the reason glycogen gets synthesized is twofold. One is it allows us to store and hide a bunch of sugar in cells, and then immediately give the cell a sugar rush by just clipping it up and saying, oh, there's the glucose. So it's hiding the glucose in an inactive state. The other thing is that it can hide a lot, because when you have two glucose molecules, the concentration of glucose contributes to the osmolarity of the cell. And if you just started storing many individual glucose, you would increase the osmolarity and make the cell really hypotonic, like that, right? So you just chain them together, because when you chain them together, it's now just one glycogen. And that's kind of a surprising, confusing reality of osmotic pressure, is that when you just change molecules together, now you just have one molecule instead of 2 or 200. Yep, that's the same reason why vitellogenin is made as a yolk protein. Instead of trying to put a lot of individual amino acids into an egg cell to store them for after fertilization to quickly build proteins for the embryo, instead of storing individual amino acids, the cell stores proteins. And the purpose of vitellogenin is just to hide lots of amino acids. And as soon as fertilization takes place, the cell, the fertilized egg cell, just starts to break it down to individual amino acids. And in doing so, it's able to contain many molecules of individual amino acids without making the osmotic concentration really high inside the cell. Cells do that a lot. Yep. Cool. Osira. Yep.
For the ligands of the tyrosine receptor kinases, for non-tyrosine receptor kinases, it's cytokine, but for the tyrosine receptor kinases, it's growth factor. Is that specific for each type of receptor?
Yes. So receptors always have very specific ligands. And when we talk about those categories, the receptor tyrosine kinases, we aren't mentioning them. But I think all of them, or maybe almost all of them, have the ligand in their name. So the insulin receptor, the PDGF receptor, the EGF receptor, and so forth. And that's true, maybe less true, but I think it's true for the non-receptor tyrosine kinases too. That the receptor, and we didn't learn the names of any of those receptors, the receptors tend to also have the ligand in their name, like the TNF receptor and things like that. Yep.
And for the SH2 domains, maybe this is too detailed, I know. For the non-tyrosine receptor kinases, it gets included after the receptor gets phosphorylated. Is that correct?
Yeah, and it's actually in the receptor tyrosine kinase pathway where we have that SH2 domain. And that is diving in deep. Where it fits in is just between the receptor tyrosine kinase, like under a scab, when the PDGF receptor gets activated. It actually binds a protein that helps go between the receptor phosphorylated domains and activating RAS. And I mentioned in class, the reason that the book kind of talks about it in a little detail is because SH2 stands for Sark homology. And remember, homology is a more general term. It means these proteins are related, but they're not orthologs or paralogs. They're just related. And there is a set of proteins that have a domain, have a region of the protein that looks a lot like that Sark oncogene that was discovered in the 1970s. But it's just not a great story for us. The Sark story is not a great story. It's in the book because back in the 1970s, that was the most famous one, right? And remember, they knew these were host genes. They didn't know what their functions were, right? So the most famous one turns out to have kind of a odd, kind of confusing role. And so we see it, but we don't worry about it because we have such a good example in RAS and such a good example in MYC. Yep. Yeah, but the people who won the Nobel Prizes for that work on retroviral oncogenes in the 1970s, they were working on Sark. Yeah, so that's why, and the author was working on RAS, right? And so the author is paying respect to the reality of their life, where they're like, oh, I know, the Sark is huge, but it just doesn't fit in as being a really cool one. Yep, Danielle?
In the phospholipase C pathway, is DAG required, or is that an option?
It's required because the phospholipid, it's a good name, lipase, cutting a lipid, right? So phospholipase is cutting a lipid, cutting a phosphorylated lipid, specifically the phospholipid that we call PIP. And it literally cuts it in two parts, and it releases two secondary messengers, the DAG, which are the hydrophobic tails, so it stays in the membrane and activates a protein kinase. So that's one way that activation occurs. Then the other, we'll say half of that phospholipid that the phospholipase cuts, the other half floats into the cell, and that's that IP3 that can help release calcium from inside the cell. Yep, so it has to be made, right? It has to be made, right? If we cut you in half, well, you'd have a head and you'd have legs. So your legs are DAG, your head and body are IP3. Yep? Yep?
This kind of builds on that one with the idea of secondary messengers. I'm confused on why the cyclic AMP is a secondary messenger, because like I always thought one had to be like outside, like talking through the membrane.
Yeah, so the secondary messengers come up, or second messengers come up because they're often taught in intravio. What a second messenger is inside the cell. So it's in the cytoplasm, and it's a small molecule. And so RAS is not considered a second messenger just because it's a big molecule. And AKT and so forth, FOXO, those aren't second messengers. So it's just, the term kind of harkens back to the original discovery of signal transduction methods, pathways, and it was really the second messengers that were discovered early, because they're easy molecules to identify, they're little, and they change their amount, like that. And so, and you could add them, right? You could buy some and just add it, like that, right? So they came up early as early stories in the pathway. The whole story, though, is that idea that nobody, I think, really ever talks about. But the idea is the second messenger comes after a first primary messenger. Nobody recalls the ligand, the primary messenger, but that's what they mean by second messenger. The first messenger, first message is the PDGF, and the surprising magic from the story is that ligand doesn't come into the cell. For most receptors, it binds outside the cell and does not come into the cell to do its job. So it must, signal something inside the cell. So that's where we get the word second messengers, and they're just small molecules, and and so it's kind of an old kind of concept, because everything on a signal transduction pathway fits the idea of a second messenger. RAS fits, but it's just a big molecule. So we just let that go. And then the other thing I want to mention about that is that's different. Like estrogen receptor. What second messenger is involved in the response to estrogen? None. Because estrogen and ligands for the nuclear receptors can come right into the cell. That's really what distinguishes the nuclear receptors from all other receptors. The nuclear receptors are not on the cell membrane, they're inside the cell, because their ligands can get inside the cell. So there's no such thing as a second messenger for estrogen or any ligand that uses a nuclear receptor. Yep. Yep.
So I have this drawing of the estrogen receptor protein binding to the estrogen response element, but I had a question about the nuclear receptor. Is that always bound to the estrogen response element? And if not, how does the protein bind there if the nuclear receptor is there, or does it bind to the nuclear receptor?
Yeah, so you're asking about a story that makes the nuclear receptor function more complicated. And we'll just tell the story because you asked it. So the introduction to nuclear receptors would be to say, even in the absence of ligand, they are doing their job. So we learned about how estrogen receptor in juvenile females or in males is always doing a job even when estrogen is absent, because it's repressing gene expression, right? So that's for sure. And then when we add estrogen, then it lets go of the histone deacetylase and adds histone acetylase. So it turns from a repressor to an activator because it binds to a different co-regulator. It binds from a co-repressor, histone deacetylase, to a co-activator. And that's for sure true, okay? So now we're just gonna add a little bit to that that makes it seem complicated, and that is that there's always some of the receptor also in the cytoplasm. And it's bound to some proteins in the cytoplasm that are helping to keep it there, right? When estrogen arrives, it typically binds to those estrogen receptor molecules first. And what really happens on the target genes is that it's not the same molecule of the estrogen receptor that was binding the histone deacetylase and lets go of that and now binds the histone acetylase. It's just an advanced detail that it's usually a different estrogen receptor molecule that replaces that one. It's the same kind of molecule, it's just an actually physically different one. See, so that confuses it. It'd be satisfying to just say, I'm here, I'm the molecule, I let go histone acetylase, I grab histone acetylase, right? That would be really satisfying. But see, even for really advanced people, they don't quite grasp that it's not exactly the same estrogen receptor molecule. What's going on with that is in the absence of estrogen, the estrogen receptor actually has a fairly weak affinity. It binds, but it's pretty weak. It doesn't have to bind very strongly because remember it has this enzymatic job, so it doesn't have to be sitting there all the time. As long as it binds every once in a while and starts acetylating histones, everything's folded up. When estrogen hormone binds, it has a higher affinity. And so that's why we get this kind of exchange of the molecules. But it's an advanced detail, and it would be fine to remember that it is just, it's just the estrogen receptor bound to the target genes all the time, and it either is activating them or repressing them, like that. It gets confusing though, because books show that, our book shows that, and people talk about that. And the reason they talk about it is because the proteins that the unliganded estrogen receptor is bound to in the cytoplasm are really cool, interesting proteins. One of them is a relative, a paralog of a heat shock protein. And heat shock proteins turn out to be fascinating proteins in what they're doing. We learn about them first in that stress response, that heat shock response. And that role is phenomenally amazing because they can refold unfolded proteins. You could cook an egg, and in theory, you just squirt some heat shock protein on it, and the egg white would turn clear again. That protein denaturation can be reversible with these helper proteins. So heat shock proteins are doing interesting jobs. So people are fascinated by it, but it does complicate for students learning it, complicates the situation. So that's what you're asking about, and just a little explanation of why is it confusing, right? And that comes up, right? Because in some other class you'd be like, the teacher's really focused on this estrogen receptor in the cytoplasm. Yeah? You want to hear another crazy thing? There's actually some estrogen receptor in the cell membrane. And I don't want that to erase your learning, but it turns out that the estrogen receptor can also work like a cell membrane receptor and actually activate signal transduction pathways. So, again, oh shoot, why did he have to say that? And it's just because, as you go forward, it was a foundation, then you can add to it like that. Here's the biology of it. For gene expression, transcription to occur, it takes a few minutes, right? You got to get things going and get it done and, get the mRNA out of the nucleus and get it translated to a protein, right? And so for almost all responses to estrogen or testosterone or so forth, it takes a while. And you're going to add an inhibitor of RNA polymerase, and it blocks the response, right? It requires gene expression. But there are a few examples of estrogen that have rapid effects within seconds, especially in the nervous system. So people are like, what the heck is going on there? And of course it's super, was super controversial back in the day and frustrating and stuff like that. But it's an add-on, right? And again, I shouldn't say it because I see a lot of students just being like, whoa, 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 right? They're just erasing all the things they learned about estrogen receptor. But you don't have to. You can take foundational knowledge and add exceptions or advanced understanding to them, and be like, cool, yeah. I'll give you a quick example of that. I teach developmental biology in the fall, and we talk a lot about certain concepts. They just are throughout the whole course. And one year we went down to the Maine Health Institute for Research, and we met with a stem cell scientist, Joe Verdi. And Joe is very contrary person. He didn't even want to look at us. He was watching a Red Sox game on his computer, and he just had his back to us in his office as we chatted with him. Yeah, he's great. Very scary for students, right? Some scientists are really, really scary people, because they seem scary. He's scary. And so I knew that this would I said to Joe, we've learned all semester, we were learning about these terms. I listed the term, and I said, what do you, as a stem cell biologist, what do you think about those terms? And he said, those terms are all horse, S-H-I-T. And the students just about fell down, But the truth is, those terms are true and real, but a lot of them get turned over in stem cell biology because you can change a cell into a stem cell and open the doors for possibilities, right? Because a lot of concepts are, well, the cell's going to differentiate into a muscle and it's not going to change, but you can change it, right? So it's like that, where more advanced knowledge is actually benefited by having a strong foundation so that you appreciate, whoa, this is really actually pretty amazing to see some very fast responses to estrogen because we know we have to look for a different pathway like that. So that's that story you asked about. And I know because I partied with this guy named Keith Yamamoto, who was really the boss of nuclear receptor biology back in the day. And he said, yeah, that's it. He explained it to me like that. Yeah, well, he's really wasted. That was great. Oh my gosh. Yeah, I know all those people just because I'm old and, I went to meetings like that, like really funny stories that we should just stop and tell. Like Jim Watson gave me high five once because of how I made a drink spill on another professor like that. I wasn't a professor. Yeah, just funny. It's just really funny, because they're all dorks, right? They're all just, it's like helpless dorks. Anyway, let's just focus on our exam. Yeah, did you have a question, Sam? And then Anna?
I do. While we're on the topic of nuclear receptors and exchanging co-regulators, our text uses the terms mediator proteins and general transcription factors. I'm wondering if you can shine some light on those terms and how they intersect.
Yeah, so we really like to emphasize transcription factors. And it turns out that when we start talking about remodeling chromatin or activating gene expression, there are other proteins that are players in the process. And it just becomes a lot to learn to try to add in these mediators that are go-betweens like that. Yeah, and maybe there would be a point or two on the exam about one of those proteins, because the book talks about them in a fair amount, but it's just not, it's like that. You see that with regard to chromatin remodeling, where we said, for our course, let's just learn 3, right? And we're not going to learn more. DNA methylation, histone acetylation, and phasing nucleosomes. Those are great. The book goes into 10 or 20 ways of modifying histones. And there are some really famous proteins. Here's a quick example that is not new learning, just to mention it. I said in the first quizzes, right, I said, what cells express MYC? And I said, for answering the quiz, it's wrong if you say a myoblast, because myoblasts are around for a long time, and they're not expressing MYC. It's only when the muscle starts to differentiate. So the best answer to that would be differentiating muscle cells. Lots of students on the quiz said muscle cells, and I gave that credit, but I don't know, because it turns out in some cases, once gene expression has begun, the same transcription factors keep it going. But then in other cases, a whole different set of proteins take over, and there are these mediators. Like one is called big group, a family of genes called polycomb, that keep going, so you can actually in some cases sustain gene expression even though the original activators are gone.
Did you mean to say Nick?
No, what did I say? When I was saying what?
He's-
In the muscle cell.
Oh, I should have said MyoD, sorry,
Okay.
yes, yeah, sorry, yeah. So, yeah, that's a good answer. If you said, oh, I think MyoD is present in muscle cells, that's a good answer, because it makes sense for where we're at. It turns out, again, in more advanced courses, yeah, sometimes it's involved in getting the gene expression going, but then it becomes less important. And so there would be examples of these that people would like to call mediators, because they're involved in gene expression and in transcription regulation, but in other kind of roles that aren't so classic as MyoD. Yeah, let's see, and then Anna, you had a question.
I do. I have a question about, so the gene amplification. So when you have multiple copies in the video, so we had multiple copies of the MYC gene, would that just be one cell? One cell had that mistake that's then gonna, be a whole system-wide problem, or is this many different? Like, I guess I didn't quite understand. I know like a gene duplication can happen, but this is like, how could it happen so many? Like, how could you get 20 different copies of MYC gene in one cell? Or is it like system-wide, like you had?
It's in one cell. And so here's something that we didn't talk about. And incidentally, when I was in college, I had a professor named George Savanich. And when he was in front of class, he would hide, literally like this. Yeah, there was different, like the podium and stuff, and he was hiding like that to talk with us. Yeah, it was super weird, because he was a psychology professor, and you're like, hey, you got some weirdness going on there, buddy. You might want to read some of the books that you're teaching from. Yeah, and he was just really, yeah. Now, all psychology professors, I don't know them that well at USM, but I had some where I went to college, and man, you're just like, wow, they're doing great. Yeah, so I'm doing that today because it just reminds me of George. You're just like, yeah. Okay, sorry. So your question was about what again?
Gene amplification, is it just one cell?
Yes,
Or
okay,
is
so
it-
here's something we haven't talked about with regard to a tumor. Here's a brain, you have a tumor, we're gonna remove that tumor, okay? So, there's the word, just to back out of it, there's a word called a clone. And you could probably know, oh, we could potentially clone you, and it would be your identical twin. So that's a clone. A clone is like a photocopy. It's the same thing over and over again, okay? So in a tumor, folks would say that tumor cells are clonal. And that means that when you look at them, they're all the same. They all have the same mutations. What has happened? And the answer is that as a tumor is growing, new mutations occur, and the new mutation occurs just in one cell, but it increases its ability, and so it's like competing against the other cells, and then as the tumor gets bigger, more and more of the cells are that cell. And then another mutation takes place in one cell, and then pretty soon all the cells are that cell because it's competitive. And you can think about it almost like evolution by natural selection, right? At first you have a variety of cells, and then one has a mutation or organisms, and then it sort of overtakes the population. And so tumors are always clonal. And so in theory, to answer your question, you could take a tumor out and you would say, early on, the tumor has what? Oh, it's clonal. And there are no mutations in the MYC gene. But this is going to progress, it's going to get worse, and the next time you look, there are 10 copies of MYC in every cell. What happened? One cell got gene duplication, and it grew to overwhelm the other cells. And then later, it could go up, and a lot of times when they remove a tumor, by the point they remove it, all the cells have 100 copies of the MYC. It was originally a single cell that had a copy, and then, took advantage of that, and then a couple copies, yeah. And we know that gene duplication is a rare mutation. One of the things that happens in cancers is they decrease their DNA repair. And they just do that because then that becomes a natural selection advantage, because they're making more weird cells, and the more weird cells you make, the more likely you are to find one that's going to grow faster and worse, troublesome, than the other cells. So that's what it's like. Is it's going to start out as just a normal MYC. One cell's going to get a couple of duplications, and it will grow faster than the others. Then one of those cells will get a couple more, and they're rare, but they become very common in cancer. So you see hundreds of copies. And what's going on there is, that you're really not changing the gene. They're just the normal gene, but you just have more copies of it. So you're making more of the MYC RNA, and it just starts to overwhelm those downstream instability strategies for keeping MYC activity low. Yep. So that's a set. Gene application is something very common in cancer, especially late stage cancers. Some gene has gone through tremendous gene duplication, and it's a problem, like that. And so it's a good example for us to learn, because it also helps us appreciate, right, that would cause trouble. If you just kept making more and more MYC genes, you're going to make more MYC protein, and it's going to overwhelm those downstream effects, right? So you could ask that about gene duplication in RAS. Do you think gene duplication in RAS is ever found to play a role in cancer? No, never, ever. Because it's not about the amount. It's about the type of protein that gets made. And so it's not, well, if you had more copies of the wild type RAS, that would be problematic. It wouldn't.
Wouldn't more RAS, though, make more MYC?
No, because there's always lots of RAS in the cell,
Okay.
but it's inactive RAS.
Oh, 'cause
And
RAS
it's
is
all about the protein, and if the protein gets messed up.
Mhm.
So the mutations had to be in the coding region of RAS. And so those are two really nice oncogenes, because they're on the same pathway, RAS and MYC, but their story in cancer is very different. It's mutations in the transcription unit only, and in the coding region only, and in certain positions of the coding region only that affect RAS. Only those. And then in MYC, it's only mutations in the regulatory region. Never in the, well, maybe they are, but really never in the transcription unit. It's not about changing MYC, it's about making too much of it. Yep. Yeah, right? I mean, it's just like you got two arms, there's two oncogenes, get some tattoos, you guys, let's go, right? I mean, really, yeah.
My next question is about beta-catenin. Do we just need to know that it's degraded if the want isn't there? Like, do we need to know anything about that specific protein, what it binds to, what it
Yep,
does?
these are, yeah, so imagine that those last examples are, individually ones, are just super famous stories in biology.
Okay.
Like beta-catenin is the transcription factors that's this target of Wnt signaling. So Wnt is a signaling molecule that goes between cells, and it binds to a receptor, frizzled, and the signal transduction pathway stabilizes beta-catenin. So it starts out as a kind of a funny story. We have lots of cells that are making beta-catenin all the time, and it just gets destroyed immediately. Isn't that a waste of time? What a weird mechanism. And then if there's Wnt, it can stabilize the beta-catenin and allow beta-catenin to do its job. Yeah? Yeah. And the reason it's in the book is because there are several proteins on that pathway. Wnt is a protein, Frizzle is a protein, and beta-catenin a protein, and the intervening proteins are also ones we don't need to know the name of. But there are several proteins, and in every animal on our planet, there are orthologs of those proteins. The Wnt signaling pathway, not just one protein, but a set of proteins is found in every animal on our planet. The Wnt signaling pathway's significance for life on Earth, the history of life on Earth, life for each of us, makes the role of hemoglobin in the story of life on Earth look like a crybaby. Wnt is just a giant figure in the history of life on Earth. It's a tool for regulating how to put animals together. It's like a tool in a toolkit. Yeah, and it's just super easy to find it in any animal. And it's the whole signaling pathway. All the proteins in the pathway have to have genes, and all the genes are just dramatic ortholog. It was invented long, long ago. And I mentioned, is Wnt found in plants? No, never. Because plants evolved multicellularity independent of animals. So there's just so many different angles why the Wnt signaling pathway is a monster. However, we've got other, we've got other signaling pathways that we want to learn a lot about, right? And so for that, if it comes up on the test, it's going to be a 2-point question. Like that. What does Wnt do? How does Wnt work? And the answer is stabilizes beta-catenin. Yeah? Like that. But just as a side note, in our bodies, it turns out that that Wnt signaling pathway does a lot of things. Sometimes it activates MYC. So Wnt signaling pathways can be involved in cancer for a whole bunch of different reasons. Here's another thing. In the bone marrow, those stem cells are listening to other cells in the environment, and they're listening to cells that are making Wnt, like that. It's just gigantically significant over and over again in your life. Like, let's say you put together a car with no screwdrivers. You don't have access to a screwdriver, right? You have the other tools, but not that one. Good luck. How far could you get, right? You put the wheels on, the rims. That's it, like that, right? You're like, not too far, right? So it's like that. So that's why they're in there. Those last ones are. They're just monumentally significant, right? There are plenty of professors that if they weren't there, they would just write the author and say, well, I'm not using your stupid book, right? But that's what they're there for. And for us, all of them are just examples of genomic signaling. And we like that. We like to see how they're activated. Not just gigantically significant. For us, the significance is it's cute. You just cut the receptor and it releases a transcription factor. It's inactive because it's stuck onto the receptor. It's not in the nucleus. But as soon as you release it, it can float into the nucleus and do its job. So it's cute. It's classic. Fun. Yay! Like that. Another tattoo. You've got legs. Right? Yeah. Yeah, so that's what it's there for, right? So we're not stacking. We talked about it pretty quickly at the end of the chapter. Our fave thing is talked about receptor tyrosine kinases. Our second favorite thing is to learn some about G protein coupled receptors. And then those are these isolated ones. Yep. Yeah, and that's all about, biology is hard because you have to get familiar with this pyramid of knowledge or a hierarchy of knowledge where there's some stuff that's more significant than others. Like that. And it's because there's a lot to learn, too much to learn, right? And that's why I use textbooks, because I'm like, struggle with the textbook, right? I don't want it to wreck your learning and like erase your learning, but it's good practice to struggle, to try to learn as much as you can. Yep. All right, other questions people have about anything at all? Yes?
So during CHIP, after you add the cross-linking agent, do you have to lyse the cell before you add the antibody?
Yeah. Yeah. Yep. So CHIP. CHIP stands for chromatin immunoprecipitation. So chromatin is DNA and all the proteins bound to it. So examples of proteins bound to DNA would be all those transcription factors and all those histones and lots more proteins like the mediators and so forth could be bound to DNA. And we're super interested in them. They're all bound to DNA by non-covalent interactions. So it's hard to study because as soon as we lyse or break open the cell, we could break some of those non-covalent binding, or we could make false new binding because we've broken the cell open. So CHIP, chromatin, we want to study the chromatin, then immunoprecipitation. So immuno is something to do with the immune system, and we can use animals like rabbits and mice and so forth to make antibodies and create tools that can allow us to bind to proteins of interest. So the example we would go to is we could take purified estrogen receptor from a source, and we could inject it to a host animal and make an antibody, and that would bind to that receptor. And then we're going to precipitate it. So immunoprecipitation, we're going to pull the antibody down, And with it, we're gonna pull its target. So we're gonna study the estrogen receptor, we've got an antibody, and we can use that antibody to pull the estrogen receptor down. The cross-linking is done when the cell's intact. The cross-link is a covalent bond. And so the first thing to do is, here's the happy cells, they're in a dish, and you just add a cross-linking agent. And randomly and very quickly, there's some covalent cross-links made. Now that allows us to lyse the cells, break them open, using a detergent that will also disrupt any non-covalent bonds. So we don't have to worry about other proteins binding to DNA incorrectly. We don't have to worry about the estrogen receptor letting go of its targets. We've physically connected them. So now we have the estrogen receptor bound to all of its target genes, and it's covalently bonded for the moment. The cell's broken open, we can add the antibody. The antibody is a protein, it's a big protein. We have to break the cell open to use it. We have this lyse cell or this lysate broken open cell, and everything's floating around in the test tube. We add the antibody, and then we pull the antibody down. Lots of times the way people do that is they will have the antibody bound to a magnetic bead. And let's just use that example, because they go through the full procedure. They make the antibody, they have it ready. Then they take the cells they're studying and they cross-link them to make covalent bonds. Then they use detergents and stuff to break it open, make sure that all non-covalent bonds are not happening. Then they add this antibody that's bound to a magnetic bead. The antibody will bind to the estrogen receptor, and the estrogen receptor is many molecules bound to all the different target DNAs. Then they'll just use a magnet to pull that whole thing down, to precipitate it, to pull it away from all the other molecules. Then they'll reverse the crosslinks, because you can reverse the crosslinks just by heating the sample. And now they have little bits of DNA that estrogen receptor was definitely bound to in vivo or in the living cell. And they just sequence those little bits of DNA. It's enough information for them to see every gene. One of the target genes is definitely going to be vitellogenic, and lots of other target genes. Yeah, so it's a super cool method because it's very convincing, and it goes to what you were asking about, is you got to lyse the cell open, break it open. But that's a kind of, in the experiment, kind of a dangerous game to play, because what if you lose the connections between the transcription factor and its target, or what if you make some incorrect false ones, right? So it's done where you kind of do a sneak attack, where you cross-link the cell when it's alive. Yeah, like that. Yeah, so it's a super powerful method, because usually the scientists would have started out with one target gene. Oh, we know the MyoD protein, transcription factor, activates myogenin. I wonder if it activates other target genes. And it does. We just learned about myogenin. It does. It activates a bunch of different things like that. Super fascinating to really learn what is its total function. Yep. Yeah, and that's one method that's really satisfying to teach and learn, because it helps you appreciate, what is how they learned about those things. And molecular biology like that is like, my advisor in grad school, John, taught a course called Inside the Nucleus. And he never would say the word nucleosome or histone, ever. Instead, in teaching the course, he only talked about the methods, right? Because even naming the proteins is kind of like, almost like being too much of a summary. So what's the data? And it was just painful. It was a grad level course, it was taught in the evening, and the undergrads that were in it were drinking shots of whiskey like that during the class. And John was such a nerdy professor, he didn't even notice that. He was just like, okay, yeah. And John also, just last point about John, because you're just like, that is sick. John's tests were multiple choice, and they were just 10 questions. 10 multiple choice questions, each was worth 10 points. Yeah, yep, right? Oh my God. Yeah. And I wanted to join John's lab, so I wanted to do good in the class, well in the class. And one of the grad students in the lab at the time, I was just trying to get in the lab, Jeff, turned around to me during that class when everybody else was drinking shots and said to me, literally this, me, poor little Dave, said to me, you can join John's lab if you believe John is God. It's just like, yikes. It was yucky. I didn't like it. Yeah, especially because I was a grad student, so I wasn't drinking shots. In the evening, it was like, it might have been Friday nights, and you're just like, yo, this is great. What a great class. Sorry, I don't know, I got on that tangent, but it was not helpful. Oh, John's lab invented chip. That's a sort of a highlight, sidelight. Yeah, pretty significant lab. Yes?
I have a question about the gel mobility shift assay. Does that it uses the DNA deletion studies basically because you are going to add the protein to the enhancer and it's just to prove that your protein's binding to your enhancer?
Yeah, so that's a good one because those are like 2-point questions, right? It's describe that method. And I've chained them together intentionally. I think all of them are in the book in chapter 7 or somewhere like that, you know, but they fit together really nicely. Because you start out, you're like, I know that, that this gene is activated by cyclic AMP. Because if I add some cyclic AMP to the cell, this gene gets activated. So I'm going to call it a cyclic AMP response gene, and I want to understand how it gets activated. So there must be an enhancer that has a transcription vector that's sensitive to cyclic AMP, right? So the first thing is to do some deletion studies. In a test tube, I'm going to take that target gene, and I'm just going to start deleting it and testing it. I'll delete a little bit, and I'll put it in the cell and test if it's still responsive to cyclic AMP. And as I delete it in these deletion studies, it'll be active until I've deleted that enhancer. And then it's like, that's where the enhancer is. But that just provides one side of the enhancer. We don't know what else is needed further on. So we start out and do some deletion studies. Then we can do some footprinting studies. And that's just, let's see where proteins are bound. So we can add this enzyme, deoxyribonuclease, DNase, that will cut in the middle, endonuclease will cut in the middle and just cut up the DNA that's exposed. The footprinting studies are because the proteins that bind the enhancer protect it from cutting. So we get a footprint where it's protected. So that's great because it gives us a limit. And enhancers, I don't think we've mentioned, they're only about 20 nucleotide letters long. They're really short, like that. So now we know where there's a footprint there and we know where there's an enhancer. So now let's use that little piece of DNA and we'll use it in gel mobility shift to see if we can find a protein that binds to it.
Oh, you're using the little piece that wasn't cut up by the endonuclease.
Mm-hmm. Yeah.
And wouldn't that also include the promoter and the transcription unit? How could you say you just have the
Yeah, because there are different proteins, so we've drawn it every time, right? The enhancer could be far away here, and the promoter is always right next to the gene. So we would see footprints made at
so there's more than
the
one
thing.
footprint.
Yeah, it could be a variety of different footprints, right? And we're interested in the one that our deletion studies pointed us to. That's the one that's really cyclic AMP dependent, right? And so then they're like, okay, so we're gonna do this gel mobility shift, and they see that, and they're saying, you know, we should name that protein because we see that active, we see that binding, and they, like I said, they say, let's call it, well, let's call the enhancer, let's call it the cyclic AMP response element. Cool name, right? Cute name, appropriate name, biochemist-based name. And we'll call this protein that binds and slows the mobility down a gel. That could be the cyclic AMP response element binding protein, or CREB. We'll just call it CREB, you know? And we just have that evidence. But we see that we can get some protein to bind in a test tube and slow the mobility down a gel. So now let's take a bunch of that DNA sequence and make a column and pass liquid over it to get the protein to bind, and then wash everything else away and release that protein that we're calling CREB and study that protein. Yep. Yeah. So and then you say, okay, we found CREB. CREB's a super cool thing. We found one target gene. I wonder how many target genes there are. So then the last one is, let's do CHIP. You know, we got the CREB protein, we purified it. Let's inject it into a little bunny, make antibody, and pull down all the targets of CREB. And under different conditions, under different cell types, right? Because the targets can vary and stuff like that, right? And so that, you know, it just fits together in a nice logical chain of discovery that people did, you know, kind of in that order. There are more examples and stuff, but I like it because it's 5 or 6 methods that you're like, okay, I got that flow. that kind of workflow. And it would have taken years and not going exactly like that, and there are other methods that people use, but that's a nice one to kind of help you see how those fit in. Yep.
Did you kind of combine the gel mobility shift assay and the affinity purification?
No, so first would be the gel mobility assay, where you're just like, we've seen the footprint there, and I wonder if we could get a protein to bind to that DNA, because it's probably that protein that makes that footprint. So the gel mobility shift is really kind of a confirmation that we can get a protein to bind. Because if it bound, it will slow the mobility down on the gel, and we can see that. It doesn't help us find what the protein is. It just confirms that we can establish the conditions that we can then use for affinity purification.
Okay.
Because you say, well, what kind of salts will we add to keep the protein bound as we wash the other ones away? And you say, well, we've tested those out in the gel mobility. Those salt conditions work in gel mobility. We want to purify that protein. Whatever it is, we think it's probably the Krebs we are seeking. Maybe some fake out thing, but, and you can optimize the conditions pretty quickly with a gel mobility assay, right? Where you can say, let's adjust the salt concentration to optimize the gel mobility shift, because that concentration of that salt is what we want to use when we do affinity purification. Yep.
Okay.
Yeah, so you can chain them together logically like that, and it kind of, like I said, like a workflow, and chip, is at the end, where you're like, oh yeah, and it's whatever, it's the estrogen receptor, or it's MyoD, or it's MYC. Yeah, and well, what are its target genes? And they are different in different conditions and different cells, and so you do chip a bunch. Yeah, and yeah, it's super cool. Yeah. And again, back to Sam's question of mediators, there are lots of stories in the biology of the regulation of transcription. But this is a good core, set to learn about the biology and also some about the discovery, because the methods are sort of self-reflexive or something like that. If you understand the method, you're like understanding the physicality of it. It's so physical. It's always physical, interactions. Yep. We're doing great. We won't have a concept chat today because we're cranking over stuff. Yep. I will just say that that old exam, lots of the questions could be the same. Not all of them, but lots of them could be the same. And so there probably will be some questions on our exam on Thursday that are just from the old exam. That would be natural. And one reason that would be natural is because I would encourage you to use that as a resource, right? Right? And I always remember in college, I had this intro bio course, and I went to college on a small college on a gigantic campus, hundreds and hundreds of acres of woods. And the first lab activity was to go out and find certain trees on campus. And none of us did it. It was just so stupid. We're like, oh, great. We go to college in a big wooded area. And then on the first exam, in the lecture class, the professor asked about every one of those trees. It was like a third of the exam. And everybody was just, oh my God, right? But see, there's some of that, and it's not mean-spirited, right? The professor's trying to teach you to use the resources that they're providing, like that, right? So think about that too. And I pulled up the old exam because I thought maybe some people would ask questions about it. What's the answer to that? That'd be good. Yeah. But anyway, look at the old exam. And you can't work them all out. You don't have an answer key, but it's good. It's good studying. Studying to, that's like people who take a practice test for the MCAT. You're gonna do that? Anybody in here ever gonna do that? Right? So you take a practice exam. It's like a practice exam to get you in the groove, and mentally thinking about how you'd answer. Yep, great. So that's a good thing to use. Yep, other questions people have about anything at all? We can just keep going. Yes, Alex?
On the first exam, there's a lot of, there's a few questions that ask to draw out the pathways. I was just wondering, to what level of detail?
Yes, the level of detail that we learned in class.
Okay.
Yep. There might be a question on the exam not to draw one of the other proteins, but to, that maybe the book described it in some detail. So that might come up, but it's less likely. And when the drawing is done, well, so like the drawing, how to do the drawing and label and drawing, just the way we did it in class. Remember that when I draw a gene now, I don't label it all the way, because I just assume you could do that really, really well, right? So the way we've learned about how to label it in class, okay? So let's just use it, for example, more specifically. So on the Wnt signaling pathway, we would like to know Wnt and beta-catenin for sure in a drawing. And then the next favorite thing would be that frizzled receptor. And then that's it.
Okay.
The signal transduction pathway includes more proteins like GSK3 and so forth and disheveled and stuff, and we just didn't need to know those.
So just like the big players, like, and then in a pathway drawing, do you want us to like also explain what is happening, like that it's
Yeah, there could be a case where I say, how does Wnt signaling pathway work? And
Yeah.
and you don't need to draw it, you need to describe it. Yep.
Okay.
Yep. And be aware that sometimes I ask you to both describe and draw and label, other times I don't. And if I don't ask you, you can include a label drawing in an explanation. But if I say explain the Wnt signaling pathway, the answer is not to draw and label it, because that's not an explanation. It can be included in an explanation, but the explanation has to include some words, describing, the key highlights. Yep. Yeah, but yeah, the pathways, the figures in the book, have a lot of detail. Yep, and it's a hierarchy of knowledge. We don't need to know all the detail. We're looking for highlights. Yep. Now, that's the Wnt signaling pathway. What if I asked you to draw the PDF3 receptor signaling pathway? Well, then we know that really well, right? Because it's got RAS, and it's got the MAP kinase players, and it's got L. And it's got a target gene MYC. We have so much to draw in that, right? So we know the details of the pathways in varying degrees, right? And it's the whole point of trying to get you to learn 1 really well, and then 3 pretty well, and then a couple more in the G protein coupled pathway, some 2, and then these others a little bit. Yep. Yeah. Yep.
Can you review, so this is from the test of this.
Yes. So there's a
Can you
good question
review
that we didn't talk about.
The significance of the sizes of
Yeah, so it's a question, and I think I have, like I said, I think I've pulled my test up. So we can scroll to this question, so you can see that, right? So it's this question, and that's something we didn't cover. This stands for the androgen receptor, and so the testosterone receptor, so not the estrogen receptor, the female vertebrate receptor, but the male vertebrate receptor. So it's the androgen receptor, and what they did is they did CHIP, and then they looked at all the enhancers that the androgen receptor binds to, and they're only about 20 letters long, and they aren't all the same letters. So in the binding of the protein to the enhancer, the place where it's going to footprint it and protect it or mobility shift it, stuff like that, not every one of the nucleotide letters is equal importance. And so what they did is they did CHIP, and they sequenced all the target genes and the enhancers, and then they made a diagram to show which ones are the most important. So the big letters mean that in almost every one of the target genes, there was a G in the second position. So it's a big, tall letter because it was really commonly that G. And so you see some big, tall letters, those are important. The spaces can be important too, but not necessarily the letters. So you'd say, why are these letters even there? And the answer is because it's the spacing, right? Because it's a protein with physically arrayed amino acids that are going to do the binding. They don't bind to every nucleotide. They bind to some with great significance and others with less significance, like that. So we didn't cover that, but it's called, this graph is called the consensus. A consensus means agreed upon binding site. And it's just showing what the consensus binding sequence would be, like that. Yep. And so you can see the first one, the A is important, but it's not always A. Or often A, but it doesn't always have to be A. Like that, yep. And so that's what that's driving at, and that's an example of something that is a topic that we just didn't cover in our course that came out that semester. Yep. Yeah, and why are some of the letters bigger than others? What part of the androgen receptor binds to these response element, and what mineral atom is required? So you could answer the second part, right? Because it's zinc and it's the DNA binding domain, right? So that can happen. I don't do that very much, where it adds a question and you're like, we didn't learn anything about that. But if you stop and think about it, you know, you're like, it's some kind of data graph that I've never seen. But you know these answers, you just don't know why some letters are bigger than others. I don't really write questions like that where I'm like, hey, you know, here's a challenging situation. I should, that'd be a good challenge, you know, but I just don't write questions like that. They're more matter of fact, like that. Yep. Other questions people have? We're doing great. So we have a couple more minutes. Yep. Yep, this, and then we'll probably have to head out. Yep.
My question about DNA looping
Yep.
is, do we just need to know that the DNA looping is what makes the enhancer only act on its certain domain? Do we need to know about like the cohesion and the other proteins?
Yeah, here's the deal with looping. And the idea is that what we talked about is that the enhancer can be far away from the start of transcription unit and the promoter.
Mhm.
But it's like a tether, like you have a dog on a leash. For the dog to be able to get to it, the leash is going to have to loop. And so for our course, we acted like the leash would just loop randomly, that you were going to increase the local concentration of the bitey dog by having it tethered. It turns out that there are some proteins that actually play the role of making a loop, right? And that just increases the, it shortens the leash, you know? Like your friend is like, oh, you have that bitey dog on a long leash, you know, you might not get bit. And then your friend grabs it and just pulls it and makes a loop. So the dog is like this far away from you. You're gonna get bit now, right? So there's some looping proteins. Who knows what the name of one is? Because that'd be a good 2-point question, because it's something the book shows,
Like
the book
cohesion?
talks about.
Like cohesion?
Yeah, so cohesion is a looping protein. And that'd be a 2-point question, or part of a 2-point question that would be good. That would be from the course, because from the book, that's not our course. And maybe it'd be 1 point. It'd be maybe how can the enhancer be so far away from the start of transcription? And the answer is, well, the DNA is like floppy, like a tether. And then it could say, and what's the role of cohesion? And you could say it's a looping protein. Or you could say, what's the name of a looping protein, right? And so that would just be 1 point that would be pushing it, right? But it's there in the book. The book talks about it. The book shows it in the figure. And you're like, oh, that's interesting. Because you can hopefully see and understand the tethering concept. You'd be like, oh, I bet a looping protein would be pretty valuable, to help, push the system, right? Because you just loop the DNA out so the dog's right at your feet, just biting. Yep, and then one more question, I think we gotta go.
So in terms of tamoxifen blocking estrogen,
Yep,
does that that results in your proteins being more folded, right? Is that-
no. So the deal with tamoxifen is it's an antagonist, which means it really binds directly to the receptor and it blocks the estrogen binding so that the receptor can't be activated. It doesn't denature proteins, it doesn't damage proteins or anything. It's just competing with the estrogen for binding. And if the estrogen can't bind, we're not going to activate. One thing that's interesting about tamoxifen is that it binds, but it actually keeps the repressor, the co-repressor present. So not only can't we bind estrogen and activate the gene, but we maintain the repressor. And that's in ER-positive cells. And I can just say that there's some people that we learned about, that are ER-negative, right? So 70% of breast cancers are ER-positive. They would be sensitive, responsive to tamoxifen. 30% are not. So doctors and scientists have studied those 30%, and they found another receptor. Now there are some few number of breast cancers that are called triple negative. Can't use it because they don't express breast cancer. Then there are a couple other nuclear receptors can't use it. Triple negative. Turns out we have a Zoom meeting here at 1 with a scientist at the University of New England School of Pharmacy who's working on an assay to help identify triple negative patients and how they're triple negative, and wants to start a biotech company. So it's cool, it's logical. And we'll talk more about triple negative because we'll talk more about cancer treatments in chapter 20. Yep. But that's what we need to know for the first exam. Tamoxifen is an antagonist, binds directly to the estrogen receptor and blocks it from activating G. Great.